In this video, we will demonstrate how to conduct a gene tagging experiment by tagging beta-actin with GFP in HEK293 adherent cells using Invitrogen CRISPR-Max transfection reagent and the Invitrogen TrueTag donor DNA kit. The first step is to design the experiment and order the required reagents via the free Invitrogen TrueDesign genome editor software. For today's experiment, we're selecting gene tagging, then the species of cells, in this case human, and then searching for our gene of interest, beta-actin. Next, you will pick the transcript of the gene you wish to target. Then, select the type of gene tag you wish to perform. For this experiment, we'll use fluorescent tag expressed gene. Select whether you want the gene tag on the N or C terminus of your gene. We'll use the N terminus. Select which fluorophore your gene should be tagged with. We'll use GFP. Lastly, TrueDesign asks what antibiotic resistant selection you wish to use to enrich for successfully transfected cells. Today, we'll choose puromycin. Select the Add Tag button to see where your insertion will be in the gene topology map. Clicking Next initiates TrueDesign's algorithm, which will scan the desired edit site region for all proto-spacer adjacent motifs or PAM sequences. It will then rank the recommended gRNAs for performance and specificity and filter out those with poor off-target prediction. If the site you are targeting lacks a PAM for CRISPR, TrueDesign will design talons to target the desired edit site. Clicking the off-target number gives you a list of the possible off-target editing sites. However, just because an off-target is listed, it does not guarantee this edit will occur, especially if your gRNA has a high performance score. Overall, a gRNA with a high score and a low off-target number is ideal. TrueDesign will add green check marks to the top gRNAs it has identified. We recommend trying 2 or 3 gRNAs for each new gene target you are editing. Clicking on each gRNA will show you where it lines up with the site of interest. With the PAM site highlighted in the purple box, simply select the gRNAs you wish to use via the checkboxes. You will notice there is a forward and reverse homology arm amplification primer for each gRNA. These are complementary homology arms for the genomic DNA location where your gene tag will insert into the genome of your cells. As you can see, TrueDesign has done the hard work of determining these sequences automatically. Clicking Next will take you to the summary page of the reagents you will need for your experiment. Simply select or deselect what you need via the checkboxes and use the drop-down menus to choose the amounts. Lastly, the software includes the option to add positive and negative control gRNAs for your experiment. These verified gRNAs are great for optimizing your experiments for a new cell type. For today's experiment, we will need gRNAs TrueCut Cas9 protein V2, CRISPR-Max, GCD primers, homology arm primers, positive, negative control gRNAs, the GFP puromycin TrueTag kit, and verification primers. Click the Download Designs and Protocols button for a summary document of everything needed for your experiment. Then click the Add to Cart button and all selected reagents will be ready for ordering. Once your reagents arrive in the lab, unbox them immediately and store them according to the package labeling. To start, it is important to perform a kill assay with a puromycin or blastocidin. This determines the optimal minimal concentration needed to kill the cells you plan to edit. Optimal concentrations are when all cells are dead on day 7 for puromycin or days 10 to 14 for blastocidin. You can measure the viability of the cells via trypan blue and cell counting or using various plate reader-based viability assays. Prior to transfecting your cells, you will also need to prepare your TrueTag donor DNA cassette. This involves PCR amplification and annealing the respective forward and reverse donor arm primers for each gRNA using the reagents included in the TrueTag kit. After the PCR reaction, clean up your PCR product with the included reagents and measure the concentration of your DNA via nanodrop or qubit assay. To start your gene tagging experiment, cells must be properly cultured, counted, and seeded to ensure a consistent and healthy cell population is ready for transfection. Plate your cells so they are approximately 30 to 70% confluent on the day of transfection, with 3 replicate wells for each control and gRNA to ensure for enough cells for downstream analysis. For this experiment, we will use a 24 well plate, plating 40,000 to 90,000 cells per well in a 0.5 milliliter complete medium. On the day of transfection, first check your cell health and for signs of contamination before proceeding. Once your cells are deemed healthy, a new gRNA needs to be resuspended in TE buffer to a concentration of 100 micromoles and stored at -20 degrees C. For each gRNA, prepare 1 RNase-free microcentrifuge tube with gRNA, TrueCut Cas9 protein, Cas9 Plus reagent, TrueTag donor DNA, and OptiMem medium. We recommend mixing Cas9 protein and gRNA at a 1:1 ratio and make sure to add the Cas9 Plus reagent to the tube last. Set these tubes aside at room temperature while you prepare a second corresponding tube for each gRNA. For the second tubes, dilute the CRISPR-Max reagent in OptiMem and incubate at room temperature for 1 minute and no more than 5 minutes. Then mix tubes 1 and 2 for each corresponding gRNA and incubate for 10 to 15 minutes at room temperature. While transfection reagents are incubating, you can optionally refresh the media on your cells to the final volume suggested in the TrueCut Cas9 protein user guide. For our 24 well plate, the volume of media will be 0.5 milliliters. After incubation, add the contents of each tube to its corresponding well of cells and place the cells back into the incubator until the next day. At 24 hours post-transfection, cells should be assessed for toxicity. Key things to look out for include cell stress, death, and contamination. If cells look stressed, refreshing the media can help them to recover. At 48 hours post-transfection, add puromycin at the optimal concentration to start selecting for the expressing cells. Over the course of 7 to 10 days, with the media being changed every 2 days with fresh antibiotics, you will start to see cells dying off while others survive. The surviving cells should have successfully received the gene tag. These images show that you will also see an increase in GFP expression over time when the cells are imaged on a fluorescent microscope. To validate the success of this knock-in, an aliquot of cells must be collected and lysed before the PCR and gel electrophoresis is performed using the pre-designed verification primers produced by TrueDesign Genome Editor software. Upon verification by junction PCR, our GFP beta-actin cell line is ready for experimental use. If you're interested in learning more about designing CRISPR gene editing experiments, be sure to check out the other videos in this series and reach out to your local Thermo Fisher technical specialist.